Welcome to section 7.1, where it's time to start talking about cells. So we've brought up cells before because they're the basis for life. When we talked about characteristics of life, what it takes to be considered alive, we came back to this idea of having cells. So in the 1800s, because we first got the idea of cells, we didn't really pin it down, but we had the general idea where we'd seen cells in the late 1700s. But it was around the mid 1800s that we really started to hammer things down and realize that there was kind of a basic unit to life, that we realized that life could only get so small. And they started to realize that some of these really little guys that they were looking at in microscopes, that they were just a single cell, whereas big organisms like us are actually comprised of what could be millions, billions, or even trillions of cells. And so they eventually stitched together what we know as the cell theory. So this is one of the major underpinnings of all of biology. Just like we have evolution, we've got the cell theory. And so the cell theory has three parts that were kind of gradually uncovered as time went on to help us appreciate why cells matter. So first of all, we have the idea that all living organisms, so everything that we're going to talk about that's life, is going to have at least one cell. Now, there's plenty of things that have many cells. That's perfectly legitimate, but you've got to have at least one for us to consider you alive. Remember, that's one of the reasons that viruses tend to get kicked out of the living category, is because they lack cells. We also have where when you look at complicated functions that many multicellular organisms have, as well as some of the functions that unicellular organisms have, single-celled, you'll see that their function and their structure comes back to cells. So we say cells are the basic unit of structure and function, or structure and organization, because when I look at something like a kidney, that kidney is just a collection of different types of cells all doing their particular job, and together they function to filter blood, which is what our kidneys do. And so when we study anatomy and physiology, our body parts as anatomy, how they work as physiology, it comes back to the cells they possess and the characteristics of those cells that they possess. Now lastly, and this one's going to be kind of interesting for later, we have the idea of where do we get cells. And this idea is cells can only come from pre-existing cells. So cells can reproduce to make more cells, but you have to have cells to get more cells. Now we're going to discuss this later because it does create this conundrum here of, well, if we need cells to get more cells, how do we get cells? We'll discuss that a little bit in terms of what might have happened to allow for that. But for right now, we see that on this planet for the last likely hundreds of millions of years, if not billions of years, the only way that we see cells is that they derive from other pre-existing cells that were already there. And the cells that come from those other cells, like the offspring that they have, tend to have the same characteristics as the parent cells that they were reproduced from. And so there's kind of this continuity where bacteria cells make similar bacterial cells. Human cells make similar human cells. And so this is one of the major underpinnings of everything as we know it in biology. So while cell theory was kind of a product of the 1800s, we've had microscopes for quite a bit longer than that. You're talking the late 1600s is where we first started to see microscopes. You can see Van Leeuwenhoek, who was one of the first guys that really came up with these, and he had some pretty decent microscopes for his time. He was great at making lenses, which were the critical piece to actually allow this to work properly, and he was using this for fabric. So he was using this to analyze fabric to see how high quality it was, how many threads there were. And so as he was making these basic microscopes, you can see this is not like some elaborate microscope here, but as he was making these, he happened to start looking at some other stuff like droplets of pond water, and he noticed what he thought were little animals and little plant cells that were kind of floating around in that pond water. And so he wrote some stuff about this, he let some other people know, and then you started to see these microscopes gain in popularity. And so over the ensuing 200 years, you saw a whole bunch of people start to kind of just poke around with this. One of these guys was an English scientist named Hooke, and he basically went through and analyzed some cork. This is dead plant cells. And he noticed these hollow little areas that reminded him of the cells monks lived in, so he said, I'm going to call these cells, whatever these structures are. And that's where we get the name cells. He's the first guy to kind of coin it so that we get this idea that there's these little pieces that are actually inside, especially multicellular organisms, that comprise us. So humans aren't just one big piece, we're essentially lots and lots and lots of these little pieces called cells. Now, eventually, we have a lot more innovations that continue on. So we have light microscopes. This is essentially just a derivation of Van Leeuwenhoek's early microscopes. And so those ones had a single lens. They used light that was provided by just daylight. And then we started to come up with what are called compound light microscopes. So these have multiple lenses. You'll see there's a lens here that's going to be called the ocular. It's the one that you look through. And then there's another lens down here called the objective. Many microscopes, you can actually rotate it, so that way you can view it at various different powers. And so the benefit of a compound microscope is the actual magnification you get is determined by multiplying the magnification of each individual lens. So if we have an ocular lens that's 10x, so it multiplies it to 10 times the size, and then you've got an objective lens that's 10x, the overall magnification we can see now is 100x, because we magnified 10 times, and then we magnified 10 more times beyond that, 100 times total. So this allowed us to get these pretty accurate, pretty close views of living organisms. And that's the main benefit of light microscopes. Because you're just shining light on them, it's not as stressful or dangerous to the organism as the other types of microscopes that we have. So we can oftentimes look at live specimens, we can oftentimes see various processes occurring in real time, as they are in living organisms, whereas most of our other types of microscopes involve us killing the organism to actually look at it. So light microscopes, while they're not that powerful, they only go up to about 1500 to 2000 magnification. If you go beyond there, you can still zoom in, but everything starts to get blurry because you have very poor resolution, and resolution is the ability for you to determine that 2 points are actually 2 separate points. And the best way I can describe this is if you see a car coming at you from far away. While it's really far away, it tends to look like it just has one headlight. At some point it gets close enough that the headlights are far enough apart that our eye can tell that they're actually 2 separate points of light and not just one big point of light. And so suddenly that car goes from looking like it has one headlight to looking like it has 2. That's the point where the resolution is functioning. With light microscopes, the resolution's not great, so if you keep magnifying, it eventually just gets a blurry image, a really close-up blurry image, but still blurry, so it's not suitable. So that's the main limitation of light microscopes, is they're not that powerful, but they are cheap, and they do allow us to look at living things. We'll go over the rest of these structures when we go through and do the microscope labs in class. Now, electron microscopes are really cool because they allow us to have way more magnification. Depending on the type of electron microscope, you're normally talking about 1 to 200,000 magnification, or in some specific types that we're not really going to get into, you can start approaching the millions. So these have much better resolution because they're using electrons, so the electrons are much easier to distinguish, so we can see things closer up. So we get tremendous magnification and tremendous detail. Now the problem with this is for most of these we end up prepping them by covering them up with a metal. And so because we cover them with a thin layer of metal for most of these types of microscopes, we end up killing them, so we can't watch things occurring inside of a cell with electron microscopes. Now there's people working on this, so this might be something in the future we can do, but for right now the normal types of electron microscopes that we're going to look at, they're going to typically kill the organism to let us look at it. And the 2 types that you'll see mentioned in your book or frequently are the scanning electron microscope, the SEM, and this one just lets you look at the surface of something. It's not quite as powerful as the transmission electron microscope. The transmission electron microscope is ultimately going to have to cut things into a thin slice, and then it can look very closely at that thin slice. So it's more powerful in terms of magnification than the scanning electron microscope, but you can only look at one thin slice, one little snapshot of that cell in the process. There's other ones called tunneling microscopes or scanning tunneling microscopes that are a little bit more involved we won't get into, but these are the 2 main types that I'll bring up. Cell types. When we talk about cells, there's actually 2 huge categories of cells, and they are very different from one another. We tend to, because we are humans, focus a lot on cells and kind of assume that everything is eukaryotic cells like us. But in reality, the vast majority of cells on this planet are going to be prokaryotic, the more archaic, if you will, these smaller, these simpler cells, because these guys can thrive. And because they're small, they can just manage to colonize things with tremendous numbers because they take up so little space. So I want to make sure when we discuss these types of things, you guys don't forget about the lowly prokaryotes because they're pretty awesome. They've been doing amazing on this planet for a lot longer than eukaryotes, and they're still doing amazing. So prokaryotes are commonly referred to as simple cells sometimes because they lack membrane-bound organelles. So this means they have no nucleus. This means they have no mitochondria. So these are some organelles you might have seen before, but they're not going to have any organelles that have a membrane that would be on the outside of the organelle. So I like to think of this where prokaryotes are a lot like a one-room apartment or a one-room schoolhouse, where they can still have lots of different areas that kind of specialize a bit, but there's not dividing walls. They're not compartmentalized. You just say my bed's in that corner, and my kitchen's in that corner, and ultimately the bathroom's in that corner, and this is where I watch TV. There's still some of these areas that can allow you to function and do a lot of different tasks, but it's not like when you have a full-fledged house, which is a lot like the eukaryotes, the more complex guys, where you say this walled-off area is the kitchen and only cooking and breaking down of stuff goes on there. And this walled off area is the TV room where I watch TV. And this walled off area is the restroom, the bedroom. And so everything's got its kind of own space that's definitely marked off and separated from other spaces. Now this can be great in terms of efficiency, but it also has huge drawbacks in terms of how much energy it uses to maintain this. If you're trying to maintain a little one-room apartment, it doesn't take much heat, it doesn't take a whole lot of electricity, all those costs are much lower because it's small and it's stripped down. Whereas if I get this big house that's got all these rooms, all those costs go up as well. So while there are some cool things to say, yes, this is just my kitchen, keep in mind there's also drawbacks. So don't tell me eukaryotes are just inherently better cells than prokaryotes. They're not. But all cells are going to have some things in common. So prokaryotes and eukaryotes will both have plasma membrane. That's just that outer membrane so that we can say this is part of the cell, this is not. They're all going to have cytosol, which is just the kind of fluid-like substance that's inside the cell. So if you kind of open it up, it's the cream filling, if you will. It's the liquid filling that's inside the cell that suspends all the organelles, suspends all the molecules. And then all of them do have some organelles, some non-membrane bound organelles. So by this, I mean things like ribosomes. You'll see all cells have ribosomes. We're also going to have certain molecules that all cells will have, things like DNA, some type of genetic information we'll find in pretty much all cells. So there will be things that all of them will have in common. The main difference that you're going to see is the idea of these membrane-bound organelles. Only eukaryotes have those.